Hi, my name is Isabella Higgins, and I'm currently an undergraduate at the University of Massachusetts. In the summer, I had the beautiful opportunity to accept the fellowship for Choose Development, and I will be showing you guys what I'm doing for my full-time weeks here at the university and what my project's all about. Over the course of the summer, I will be working in Bartlett Lab. Bartlett Lab's main focus is evolution and development in plant biology, specifically in maize, but my project will be a little different and be focusing on a close relative of pearl millet. Here is a short clip of me walking to lab from the UMass library to go do some lab work for the day. And here are a few clips of the Bartlett lab where most of my project will be taking place. The overall focus of my project is going to be on the early development of the inflorescence comparing between undomesticated and domesticated pearl millet. Pearl millet is considered an orphan crop. An orphan crop crops are typically not traded internationally. These crops receive less attention in research and agricultural training, and pearl millet is typically grown in sub-Saharan Africa and Asia. The room I am showing right now is where we keep the SEM machine, which is a scanning electron microscope. This is where I will be doing most of the imaging for this project. The SEM machine allows me to get beautiful images of the meristem inflorescence at different stages of development. This machine will help further my understanding in the structure of the inflorescence meristem inside of the plant. The College of Natural Sciences greenhouse is where I'm keeping most of my pearl millet plants while waiting for them to grow and reach different stages of development. To further look at the differences in development between domesticated and undomesticated pearl millet, I will be using multiple methods of imaging. At each stage of development, I will be using an X-ray computed tomography and then fixing the inflorescence primordia for sectioning and staining. These two different methods allow me to measure pearl millet's inflorescence meristem size precisely for further data. The inflorescence inside of a plant plays a major role of development and overall flowering of the plant. And this image here shows a shoot apical meristem. This is a group of undifferentiated cells or meristematic cells that are capable of cell division. The apical meristem is an important factor when thinking about the developmental differences between domesticated and undomesticated pearl millet. Here are some images of my favorite pictures I've taken on the SEM. In this photo, you can see the difference between domesticated and undomesticated pearl millet inflorescence. On the far right, you can see the domesticated pearl millet inflorescence, and on the far left, you can see the domesticated pearl millet inflorescence. After that brief introduction for what exactly I'm going to be doing over the summer in lab, the overall goal for my project is to determine what developmental differences between domesticated and undomesticated pearl millet that makes domesticated pearl millet be larger in length and in width when compared to undomesticated pearl millet. So we have a couple reasonings behind the morphological differences between domesticated and undomesticated pearl millet. One of them is that domesticated pearl millet starts off with a larger cluster of stem cells at the meristem than undomesticated pearl millet. Another one is that domesticated pearl millet may just simply develop faster than undomesticated pearl millet. And finally, both of these options are both possible, and over the course of the summer, I will be trying to answer these questions. Finally, I would like to thank everyone in the Society of Developmental Biology and people a part of the Choose Development Fellowship for allowing me to have this opportunity over the summer, and I would like to thank everyone at Bartlett Lab for all the mentorship and help that they are providing me for my project. Overall, thank you.